Good afternoon, good morning, good evening, everyone. Welcome to the City Earth Data webinar. Today we have our speaker, Henrik Drake, speaking from Linnaeus University, Sweden. So Henrik is interested in the deep subsurface life. He, as I have, was going to present this work. This work is more correlated with the thermochronological study, but he has also interest in the other relevant topics like the generation of methane in the deep subsurface. That is, to my interest, that could be connected, is connected to the origin of life. So it's a very interesting topic that Henrik is going to speak today. And as we authorized by Henrik that we could record this talk and post on our YouTube channel to spread the science. And also for people who missed the talk in our webinar series could go back to and watch this video. So with that, I'll give the rest time for Henrik.
Thank you very much. And thank you for inviting me to give this talk. It's I think my first webinar for a Chinese university yet. I've been to China one time to have a talk in Kunming, as I remember, but it was a long time ago. So happy to give this talk. And I'm a geologist actually from my training, but then moved more and more into microbiology. But basically I'm somewhere in between microbiology and geology. And on this first slide, you can see a part of Sweden that it looks like most parts of Sweden actually, and has like pine tree forests and some outcrops. And behind, at the bottom here, you can see actually a mid-term storage for nuclear waste. And in the red is an area that was investigated for a potential repository for nuclear waste. And this is what I've been studying a lot. And this area does not look so interesting on the surface, but when we start to dig down into the rock with doing core boreholes and also tunnels and so on, we see these fractures that lead water. And in these fractures, there are signatures, or there are actually living microbes at the moment to great depth. And microbiologists have been looking into metagenomic studies and found out that there are all three domains of life are present and active today. Then what I do mostly is to look for signatures of life from ancient times, and then looking into different biosignatures, it's called, such as carbon and sulfur isotopes, as well as looking into different morphological features. And I will talk more about that, and then we will talk about how long has actually life been present, or how long has there been habitable conditions in the deep subsurface. And then we need to go into the thermochronology perspective of the deep biosphere. So starting off first with some deep habitats on Earth. There are different habitats where there is life at present. And this was outlined in a paper by T.C. Onstott et al. About that these environments are also of interest for exploration of the other planets, such as Mars. And we are focusing on this environment here today, groundwater systems in fractures in continents. And there are some important implications of these studies. One is the origin of life and of evolution perspective, because in these environments, there are really primitive, ancient metabolisms. These are chemolytic autotrophic metabolisms that go back to the earliest life forms on Earth as we think. And they are anaerobic, and they are living in an environment with no oxygen, no light, or that looks a bit more like the ancient Earth as we think of it. And then there are some astrobiological analogs to this as well, because if we look at Mars, when Mars and Earth were similar, this was a time when life started to evolve on Earth. So that's why it's important to look back and look at these chemolytic autotrophic microorganisms that lived on Earth at that time. And then there is also an interest in underground environments on these planets with no surface waters, because there might be water down there, and there also is no radiation at a great depth, which radiation can destroy these organic molecules. So this is a good system to look for on Earth, to find out how we can detect them and so on Earth. What does these systems look like, and how long have they been there, and take this knowledge to other planets potentially. So if we look at the classic wheel of life, most of this, if we started 4.6 billion years ago, we get some first signatures and indications of life around maybe 3.5 or so. And these are from fluid inclusions that have methanogenic signatures. And there are also these molecular datings that suggest that actually microbial methanogenesis probably was one of the earliest metabolisms on Earth. And then as we move forward, we get into the great oxidation event, and then we get multicellular life and eukaryotes, and move into the times that we know better with the colonization of land and with the dinosaurs and so on. But actually, if we look on what we have today, deep down in the crust, most of bacteria and archaea, according to this study in PNAS, most of bacteria and archaea are probably in the deep subsurface compared to marine and terrestrial, whereas all of these plants, fungi, protists, and animals are completely dominated in superficial environments. And if we look back in time, there are these models that suggest that before land plants took over in terms of biomass, which is completely dominating now, it is suggested that deep environments, deep continentals and subsea floor held most of Earth's biomass. So most of this graph, wheel of life, is actually from superficial or marine seabed environments. Whereas deep down in the crust, there is very scarce or actually no knowledge of when life colonized these deep subsurface settings, and what kind of metabolisms that dominated and for how long it has been like this. And there also, I should mention that there are these intriguing findings of extremely old waters deep down in the continental crust, such as in the Canadian Shield, where a group of researchers from Toronto, for instance, and Oxford, I think, or Cambridge, they have this 1.5 billion year old waters that they found. And they also find these, that there is life forms in these waters. So it could be actually, or we don't really know for how long time have these microbes been living in isolation in these extremely old waters. And then we need also a thermochronology perspective to understand how long time has this environment been habitable. So what I will talk about is a bit about life at depth in Precambrian cratons at the moment, by looking into what is living there today in tunnel systems and in boreholes. Then I will look into this topic of deep life in deep time by looking at the rock record, mainly fractures in crystalline bedrock, to find out about biosignatures, 3 types: isotopic, organic molecules, and morphological biosignatures. Then connect it to geochronology, so-called hot moments. When have there been formation of these biosignatures? At what occasions? It seems to be episodic. And then adding the thermochronology perspective, as we have a paper that came out in this autumn, last autumn, about this perspective, about how long may microbes actually have lived down in the cratonic rocks that we have on surface today on the planet Earth, and where can we actually find the longest records of habitable conditions on Earth. So then we look into the Precambrian Shields, life at depth in the Precambrian Shields, and most of the crust is actually by volume is composed of these Precambrian rocks, but at the moment about 15 to 20% are exposed only, because there are sedimentary covers on top of them. So one is of these cratonic shields is the Baltic Shield or Fennoscandian Shield, where I'm right now in the southern part, but this is where I've done most of my studies, because this is where I have my field sites. But there is also a Canadian shield, Pilbara shield. There are important cap wall shield, for instance, in South Africa. So a lot of different Precambrian shields exist on Earth, on each continent actually. So in Sweden, we have this hard rock laboratory called ESPE Hard Rock Laboratory that is owned by Swedish Nuclear Fuel and Waste Management. There's been a lot of these microbiology deep biosphere studies done there in waters. And I'm not a microbiologist myself, but I've been connected to these studies to some degree. And if we just make a summary of the main findings and what the environment looks like down there, is that we have all three domains of life present, basically bacteria, archaea, and fungi, mostly, I guess, and some protists. And then this environment is dark and anaerobic. At shallow depth, there are heterotrophic processes that dominate, and with depth, the dissolved organic carbon decreases, cell number decreases, temperature increases, and life can persist until the temperature reaches the highest temperatures that life can persist. That is 122, as we know it at the moment. And then there is ultra small biomass, small cell sizes, and the microbial populations they lack surface representatives, most of them actually. And with depth, these chemolithoautotrophic processes start to dominate, and they are depending instead of gases that might come from Earth's interior, such as methane, hydrogen gas, and CO2. And then we have these processes, Methanogenesis, that can be both autotrophic and acetoplastic. We have sulfate reduction, nitrate reduction, and with depth, bacteria dominates, and archaea also is mostly, also the proportion of archaea might be increasing with depth. If we look at the other cratons, such as the Canadian Shield, we've been doing a lot of works with Barbara Sherwood-Lawler's group, have done a lot of work in Kidd Creek mine, down to 2.4 kilometers, finding these ancient waters that I talked about earlier with life, signatures of life in them. A lot of work by T.C. Onstad's group in South Africa has been done that find traces of life down to 3-4 kilometers. And also some in Finland. It's kind of similar as has been done in Sweden. Also a lot related to nuclear waste repositories. So if we go down into the deep life in deep time perspective, we are looking at the drill cores mainly, and we are looking at fractures. There are fractures, about 1 fracture per meter in these rocks. Closer to the surface, there are more fractures, maybe 2-3 per meter. But in some of them, we can find these kind of crystals, vugs with crystals. And when we polish them to get a cross section, we can actually find these kind of these growth zones that might be up to 10 in numbers, and these reflect different environments, different geochemical environments, and might also reflect different microbial episodic processes that lead to precipitation, in this case into calcium carbonate. And unlike sedimentary rocks, life signatures here, they will not reflect the age of the rock because they are in a fracture. In this case, the rock is 1.8 billion years old, and the fracture might actually be 10 million or 100 million. We need to get geochronologic constraints on the very part of the crystals that we are interested in. So we need to do a lot of in situ analysis and kind of do that much conventional bulk analysis work. Then we have these different criteria for biogenicity. Basically, the best is to have as many criteria as possible fulfilled. And one of them is morphology, that we look for something that looks like a microbe or looks like fossilized fungal hyphae, such as in this slide here, or this is actually a synchrotron radiation X-ray tomography from a seamount where we have fossilized fungi together with prokaryotes that looks like this. So we'll use a lot of synchrotron facilities to do this kind of renderings. This is not so common to find, these kind of signatures. It seems to be more common in the basaltic rock on subsea floor rocks than in granites. What we also do is to find preserved organic molecules. This is a classic that is used to find the earliest evidence of life on Earth. Also, and what we look for are, for instance, fatty acids or different hydrocarbons and their isotopes to get a signature of something that is organic and not abiotic, something that is microbial. So we can take the vein, either we dissolve it and get GCMS chromatograms, or we can do time-of-flight sims. We have done both. This is more of a surface technique that we can go in situ with. So getting these peaks for different fatty acids, for instance, that are diagnostic for different metabolisms, more or less. What I've used the most is actually isotopic fingerprints, or some call them chemofossils. And these are, as I showed earlier, these kind of crystals, and we handpick them from these voids or from these veins. And what we are mostly looking for is either pyrite that has iron and sulfur, of course, and they have these different stable isotopes that we are interested in, 34 and 32, mainly in these systems. We can also look for a mass independent fractionation if they are really old. But in these cases, we usually look at 34, delta 34S. And as there is this well-known discrimination against sulfur 34 during microbial sulfate reduction, and there are also different fractionations of various steps, we can use the mineral, the product in the end, that is pyrite, to get a knowledge of if there has been microbial sulfate reduction or not. If this is very depleted in sulfur 34. But with time, or if there is a reservoir effect, it can of course be a Rayleigh distillation where we end up with extremely heavy pyrite in the last portion, so-called super heavy pyrite. This is well known from different periods of sedimentary rocks and might reflect different periods of oxygenation of oceans in that perspective. But in this case, we are not looking at that. We are looking at purely deep systems within the crust. Then there is also calcite. This is the other important mineral that we are looking at. And then we have the carbon, preferentially the carbon here with the two stable isotopes 13 and 12. And we are looking at carbon, delta carbon 13, which can give us diagnostic information of different metabolic pathways, such as different methanogenesis pathways, because they have different fractionations, just as sulfate reduction had different fractionations in different pathways. So we can use this to distinguish between carbonate reduction pathway of methanogenesis versus methyl fermentation that has a higher carbon 13. And if the values, and this gets into calcite, and we can use this calcite composition to get a feeling for what is actually the source of the carbon. And if the carbon in calcite is very, very heavy instead, carbon 13 rich, it can mean that methane has formed in situ and left behind a CO2 that is carbon 13 rich. When we have done that, we can go into radioisotopic dating. And what we use the most is uranium lead dating of carbonate, calcite, and in situ rubidium strontium dating of potassium rich minerals paired with calcite. So I will show some examples here. We have used, for instance, these what looks like microbial mats in fractures that are clay minerals that has 1% potassium in this case. So we could date these different spots here together with calcite that is in the same growth zone. And calcite is microbial in composition. There is pyrite with sulfate reduction signatures. And in this case, we got an age of 393 plus minus 15 million years. On the right side is a calcite crystal that has grown from left to right. And here are different spots that reflect a different age. This is done by Nick Roberts at BGS. So he has helped me a lot with doing the geochronology of calcite, whereas this has been done at University of Gothenburg together with my PhD student Mikkel Tillberg and Thomas Zack, who is lab manager there. So if we move into case studies of what we can find in the crust, we had this study in 2015 in Nature Communications that we use a lot of drill holes from this area that I showed in the first slide, the Laxemar area in Sweden, took crystals from a lot of depth down from the surface down to 1,000 meters and analyzed them within the crystal cross-section. And what we then found was these extremely depleted carbon-13, delta carbon-13 values down to minus 125, which to my knowledge still is the lightest or lowest value ever recorded in calcium carbonate on Earth or on any planet, actually. But this reflects anaerobic oxidation of methane. It cannot be anything else, really. And it's also a methane that probably is microbial in origin. And below that, below this zone of 200 to 750 meters, there are no such values. So it might be, these are different boreholes, but it might reflect a sulfate methane transition zone. But perhaps that we are also having the heavy values here reflects methanogenesis. So it might be even a reverse sulfate methane transition zone. As the sulfate values in the waters increase with depth in contrast to in marine sediments, where the sulfate decrease with depth and methane increase with depth. When we look into the crystal themselves, we can also find these kind of trends, low values in the core of the crystals, minus 120, and then replaced in the outer part by heavier values. And at the same time, oxygen isotopes drops. These are more marine values, dropping to fresh or glacial values that then change the metabolisms. So here we can have methane oxidation that is replaced by a later metabolism that might be sulfate reduction in this case, or some kind of organoclastic metabolism at least. We also have these organic compounds within these carbonates that are specific for sulfate reduction. So it's what we think this is a sulfate dependent oxidation of methane at anaerobic conditions. I won't go into all these details of what kind of organic molecules we found, but they are there. We didn't find any specific for anaerobic methane methanotrophs though, but we found for the sulfate reducing partner. This is the usual case actually. We find more evidence of sulfate reduction in the organic molecules than of archaea. We also find a lot of places. We have analyzed a lot of different deep mines and deep boreholes in Sweden and found these signatures in most of the areas. So it seems that this is a common signatures to find deep within Proterozoic rocks like these. We also have these spikes sometimes that we have different occasions, such as spot number 6 and 10 in this case have depleted values that are methane oxidation related, whereas the others are not. So we have with time in a crystal like this, almost like tree rings, but in crystals of calcium carbonate, we can actually have different metabolisms reflected. And if we go into fluid inclusions of these, we can find that they are more brine type in the beginning when there was underneath a sedimentary cover, and with time they get more low temperature and lower salinities as well. And this is more organic activity or a microbial activity in these when the temperatures are better. Then we did dating of the first part of the system and got ages of 400 million years. And in the outer part, where most of the microbial activities, they were Jurassic in this time. So this is a lot younger than the rock that is 1.8 billion years old. So this seems to be periods where we have better conditions for life. And I will talk about the thermochronology perspective of this soon. And when we compile all of this, all of the studies we have from Sweden, we can find that we have extreme ranges in carbon isotopes, down from -125 up to +37 per mil. That is among, I think, also the heaviest isotope composition on Earth, as I know it. And these large ranges, they occur down to 800 meters and then they stop. So it seems that at these times when these microbes lived, the conditions were suitable in the upper almost 900 meters, and then that they were not suitable anymore. And if we look into the signatures of microbial sulfate reduction or bacterial sulfate reduction, we have used a lot of both the SIMS analysis or spot analysis, but also ion imaging with SIMS, that we can do line scan. And this is a collage of 4 ion images on a crystal. So there is a SEM image in the bottom, and then I draped them with ion imaging from SIMS that reflects sulfur isotope composition. And then we normalized these line scans to spot analysis. And we can see from the core of the crystal, it's quite depleted at -20, -30. And as we go further out, it increases, then drops again, then increases, and then becomes stable more or less with growth in this green area. And then it drops in the outer part. So this reflects different fluid packages that have come into this almost closed system and then being depleted almost completely. So we get this Rayleigh distillation with very heavy values up to 130 per mil in this case. And then it seems that here we've reached some kind of steady state with flow. So we can explain it with some kind of transport model that we have also probably some precipitation of pyrite somewhere else in the system to get these extremely heavy values. So taking all this together, we go from -54 up to +147. This was presented in our last year's study in Communications, Earth and Environment, a study where we got this extremely heavy value. And this reflects a Rayleigh distillation and a microbial sulfate reduction. And again, we see that these values of extreme variability goes down to 900 meters. And at great depth, we don't have them. Probably it's been too hot at that time. And they go down to -54. And we think we have barytes that are at +20 that reflects the initial sulfate of the system. This means that we could have +70 or more per mil fractionation or isotope enrichment to get to these values. And then if we have closed systems, we can drive them up to these extreme values with time. We also have these fatty acids within carbonates associated with these pyrites that are specific or more or less specific for sulfate reduction, and also isotope composition, compound specific of them, that are also reflecting a depletion due to microbial utilization of organic matter. What we did was also to, in an experiment, a natural experiment, we took out equipment from a borehole that had been sitting in the setting for, I think, 17 years. And we found these pyrite precipitates on the borehole equipment, and we knew that the sulfur isotope composition of the borehole water, or the natural fracture water, was +24. the pyrite, the lowermost one we found, was -47, which gave us then an epsilon 34s of 71 per mil. That is actually fitting to the pyrite that we found in the fractures and this very high fractionation that we anticipated. And the increase in a single crystal can be up to 60 per mil over a couple of 10 micrometers here. So they are extremely small microscale processes in these systems. Then we can also look into these, again, these calcites that I showed previously. And when we have coupled here dating and SIMS analysis of carbon isotopes. So the numbers here, they reflect carbon isotope values and oxygen isotope values in the different overgrowths, 1, 2, 3, 4, 5, that you see here growing from left to right. We have these heavy values here reflecting methanogenesis. And this zone is actually the green here. So this is dated to 35 million years ago. Whereas the outermost zone, that is reflecting sulfate reduction because it's intergrown with pyrite, it is more in the range of 15. So this outer zone has grown for maybe 10 million years. And it's kind of intriguing that we can actually see when the metabolisms have actually changed in the fracture system. And then this was all signatures of prokaryotes. So what about fossilized eukaryotes, the third part of the tree of life? And then I will just share a couple of slides of these intriguing findings of fossilized fungal hyphae that we have found, for instance, in Laxemar at 740 meters depth, that are like, it's like in a pocket in this quartz vein. We found this mycelium of fossilized or partly fossilized to clay and partly organically preserved but dead since long. We don't know how long, but definitely not living. And frozen in time, this mycelium together with zeolites and calcites and pyrites. And the pyrites, we analyzed them with SIMS. They are very small, but we could do small spots in them and got depleted values that gives us the confirmation that they are microbial and they are sitting on top of the fungal hyphae. So we speculate, or we have this hypothesis that they are in a syntropic relation where a fungal hyphae, the fungi can live at this depth by degrading a biofilm. It grows from a microbial biofilm and it's a heterotroph, so it needs organic carbon, then produces hydrogen gas that the sulfate reducers can use. And the sulfate reducers can also utilize perhaps the biomass of the hyphae. So it's an intriguing system there. And we have also found the same at depth in the Siljan impact structure in Sweden, the largest impact structure in Europe, actually. It's 380 million years old. And in pockets and voids, we found calcite crystals that have methanogenic signatures. They are heavy in isotope composition, and they sit together with these fungal hyphae that are partly fossilized to clay minerals. They are definitely not modern contaminations, and they have calcite precipitates on them, and the calcites have a heavy isotope composition that reflects methanogenesis. We could date this to an age of 40 million years, so much younger than the impact, but still the first, I think, geochronology dating of deep biosphere fungi, and also a first fossilized finding of a syntrophic relation between methanogens and the hyphae, the same kind of syntrophic relation that we have in rumens of cattle. Let's see here. And so what the ages we get, as you could see in my previous slides, they are in the area of 400 million years. So the rocks in this area formed 1.8 billion years ago. So we have also different veins that are older, but they don't have biosignatures. So this gave me this idea that why do we only have these young ages, relatively young ages, and some different also windows here or gaps that we cannot explain. So I wanted to look into how long time actually may microbes have lived in these systems in isolation. And then we come into thermochronology that has not been that much coupled to the ancient life in crust previously, which was quite surprising for me, but because it's so natural to think of, you need to think about habitability, you need to think about temperatures. And there are argon-argon thermochronology, for instance, these closure temperatures are quite high, at least for hornblende and micas. But as we go into uranium, thorium, helium techniques, we have apatite, zircon, titanite, then we are getting into temperatures that are in more or less in the range of the limits of life at 122 degrees. And then these come into play. So we can use data, or we can use thermochronology to get an idea about habitability. So we did first a study together with Willy Günther and Pete Reiners in the US, in Arizona and Illinois, and gave them samples from the Fennoscandian shield, and they did this uranium, thorium, helium of apatite, titanite, and zircon. And of course, a lot of modeling, forward modeling, for instance. And I won't go into these details of how this is done, but I will go into details of the outcome in plot. So this is the temperature time diagram of southeastern Sweden. We start here with rock formation. So the rocks are formed in eastern Sweden at high temperatures, and with time, these rocks are eroded and uplifted. At 1 billion years ago, we got the Norwegian orogeny in western Sweden. We were almost at the surface at that time. Then we got a foreland basin afterwards that depressed eastern part of Sweden to depths of maybe 8 kilometers at most. And after that, we again got this erosion. So this is like a zigzag pattern, how the current rock block that we have at the surface have moved up and down into the crust over eons. So we got erosion and uplift up to, again, almost surface, and had platform sediments on top of the whole Fennoscandian shield almost. Then we had a Caledonian orogeny that is also found in the US and Greenland. And after that, again, a foreland basin that depressed the rocks to high temperatures. And after that, erosion and uplift. And now we have this barren ground with outcrops without any sedimentary cover that I showed in the first slide. So we can just take, very simple, just take the temperature here and call this inhabitable, because it's too high for life, and habitable. And we also need to think that fluids in a fracture can have a different temperature, a bit different temperature than the rock itself. So we also use fluid inclusions of the veins to match this, like this, as I said. So in this recent paper in PNAS, I got a lot of help from Pete Reiners in Arizona to iron out my idea about using thermochronology on the deep time evolution of the deep biosphere. So this is basically the main plot from this paper that shows red, green, and yellow that are habitable in green. And this is the best fit, is the black. And the red and yellow and green are the full model. And the red areas are not habitable because they are too hot. The yellow are borderline. And then we used fluid inclusion data as well as geochronology of veins that are in orange if they don't have any biosignatures, and in blue if they have. And most of them, as I showed previously, they are in this area here, very young compared to the rock. And this is because it's habitable. The blue, they are on the border here to a little bit more reddish. Could be that the model is not perfect, or that it could be very local temperatures in the veins and so on. But they are still in this green portion here before we get too hot, and then comes again these orange that have no biosignatures. So this reflects different states when we have been at different depths in the crust. In the beginning, the rock portion that we are looking at the surface now was very deep, several hundred degrees, and we had no biosignatures in the veins. But as we got into these areas, we got sedimentary cover, and we got a lot of hydrocarbons and black shales, expulsion of bitumens down into the basement, and we started to get biosignatures, carbon isotope signatures in the veins. But the temperatures were still quite high in brines that are not the best for microbial activity. And it was only when we got here, when the sedimentary cover was starting to be eroded, and we got fresh water coming into the system, that we got these extreme isotope signatures in the vein minerals and fluid inclusions that are below 50. So this was when we got the real blooms in microbial activity in southeastern Fennoscandian Shield. If we look more in the very eastern part of Fennoscandian Shield, we actually don't have these dips below 110 because we are too far away from the Caledonian orogeny. So the sedimentary covers were thicker on Sweden than on Finland. So this is an interesting area to look for ancient biosignatures. And if we look into Canadian crust, where they have these extremely old waters, 1.5 billion year old waters, we also applied the same thermochronology techniques to these areas in the Canadian Shield up here. Here's Kidd Creek Mine, that they have found these extremely interesting ages and microbial communities at depth. And we used some data from published works from, for instance, from Kaylin McDonnell, that has done a lot of thermochronology. What is not existing in Canada are biosignatures and no geochronology of veins or anything. So we cannot do the same as in Sweden, but we can at least get the thermochronology background or framework. And if we look at, for instance, Superior 70 sample and go back in time, it takes us to 600 or 700 until we reach high temperatures that are not able to sustain microbial communities. Sorry, and at least at 1 billion years ago, it was too hot. So these waters that are 1.5 billion years old, they have been too hot for most of the lifetime. Only maybe here in the window, and at least after 700 million years, they have been cool enough to have microbial activity. And some others, we have actually plotted all of these in the paper. So if you want to have a look at them, you can look into the paper. But another one goes back to 700 million years, then it was too hot. So it's important to have a thermochronology perspective to know when has there been habitable conditions. And this will be different in different parts of cratons, as I showed in the Fennoscandian Shield, and it will be different in different cratons. So what we did was also this global, for most of the shields, not all of the shields, but for most of them, we did these plots of habitability records. And we have the Swedish case here with these zones of gaps due to sedimentary covers over foreland basins that depressed the rock. This is very common to find. And when we are looking into the longest records, we have them in this southeastern Finland, Superior 70 maybe, depending on this part here, if it's been too hot or not. And then eastern Greenland at high elevations. At low elevations close to the sea floor or to the sea, there are much shorter records because there's been so much exhumation on Greenland, late exhumation. But at high elevations, there can be these records of up to 1 billion. So to conclude, we can say that the records of habitability have been up to 1 billion years in the crust that we walk on today on Earth. And then there are these interesting gaps here and there that reflect the more or less sterilization of the rock volume. So some concluding remarks to my talk. These ancient biosignatures, we can find them of all three domains of life at great depth, and we can find, and they match with those that we find in waters living today, all these three domains of life, even fungi. But fungi seems to be best preserved as fossils, and organic molecules are mostly for bacteria, but we have a lot of methane oxidation, methane formation signatures that are probably from archaea. The isotope values in these systems are the most extreme yet found on Earth as we know it, but we might find it in other environments as well. But this could be due to that there's extreme energy depletion, so they use the easiest way, and the easiest way might be to use the light isotope, and then with time it can lead to really a lot of reuse and cryptic cycles, and in the end ending up with a very heavy isotope composition as well. We have this thermochronology that provides a roadmap to history of habitable conditions in these cratons, and that habitable conditions date back up to 1 billion years, but sometimes only to 50, 300 or so, such as in Sweden. It goes back 300 million years, then it becomes too hot. And this agrees with the dated biosignatures that we have. So with that, I would like to thank you for listening to this talk and for inviting me, and also thanks to these funders that have funded these projects that we are looking at. Thank you.
Okay, thank you very much, Henrik. And now it's time for questions from our participants. So if you have a question, feel free to open your mic, or you could also leave a question in the chat and I could read the question on your behalf. Okay, I could start with a question. It's related to your talk. Actually, you also showed that with the samples from the Kola, I suppose that's the deep drilling from Kola Peninsula for some of the samples that showing the, could you go back to that slide? Where,
yeah, I have both this one here, and I have the sample sites here up here. So these are published works by the name I forget, some Russian guy, Veselinsky, something.
Okay. Okay. So did they find biosignatures in the Kola?
Not looked at in Kola Peninsula. I have not seen any works of biosignatures. I only see these that are thermochronology. So this the only thing that exists from Kola, as I know, from the crust, deep crust, is this thermochronology that looks the same as in southern Sweden, as you can see here. So it has these gaps due to that they are close to this Caledonian, orogeny, and foreland basin. So if we're looking for biosignatures, I think we should find the same as we found in southern Sweden. This is what I anticipate. So if we go, or if someone goes to the Kola deep borehole, I think they will find also that a lot of them biosignatures are young or in this portion of time.
Okay, because in my, I'm not a microbiologist, but in my recollection, for example, for deep drilling in China, in Donghai, the 5-kilometer drilling that people did find active life by Hailiang, I think back then. So my next question is that how deep would you expect life could exist? I mean, even especially like I said, because for the places with drilling is just we only have a few places in the world that we did.
Exactly.
So
Yeah,
you could make the guess, or you could argue about that. Where is the deepest life that are coming from on this planet?
Yeah, that can be done, and that depends on the geothermal gradient. In some areas there is a larger geothermal gradient, and some areas are very low, like in craters could be even 10, 11, 12 degrees per kilometer, and that means you can get almost 10 kilometers deep life at those sites. But if you have 35 degrees C per kilometer, you cannot get life very deep, such as in sedimentary basins. It's a higher geothermal gradient, you cannot get life that deep. So I would say 10 kilometers would be the maximum in those cases. So that we have very low.
Okay, if that's the case, then Kola Peninsula, for some of the samples that they get, because Kola Peninsula, they get all the drilling all the way that's 12 kilometers, then there is a possibility that we might find very deep life signature from what they
there is, and it could be, depending on I don't know the geothermal gradient there, but it could be. But it also is difficult with the contamination issues of such a deep borehole, and that you need maybe also drilling muds that could be with oil, such as in Siljan in Sweden, they drilled to 6 kilometers, I think, in the 80s. They have a lot of problems with that they introduced oil in the drilling. And when you introduce oil, you also introduce microbes and also fuel for microbes. And you get mixture of fluids from different depths, perhaps. So therefore, it's good to also look into the veins, because these are systems that cannot be contaminated because everything is stuck within a mineral. So it's good to complete microbiology investigations with these kind of ancient or rock record investigations of fracture coatings that I do. But not many people have done it, and I think it's because the geochronology framework has not been as good as it's been in the recent years with the advent of uranium lead dating of calcite in veins. It's not now everyone is doing it. Rubidium strontium is now also available. So the first 10 years of my research career, we could not date anything. We didn't know that much. And then it's not so attractive when you don't really know what you're looking at. But when you get an age to it, it's much more easy to interpret the history of what you're looking at. Because a fracture, it can be very tricky to know when it formed compared to a rock bedding in a sedimentary rock. It's much more easy.
Okay. Okay. Thanks for that. Actually, I have several very curious questions in my own mind. And for other participants, you have questions, feel free to open your mic or directly leave a message. Okay, I will continue with my second one. This is another question completely out of curiosity. You showed the fungi and possibly some other bacteria. So I'm just curious that is there any virus in the deep subsurface?
Yeah, there is. But it's not in my expertise. But they found it also in Hard Rock Laboratory. They've done a lot of studies on viruses.
Okay, so, and that could also cause, like, say, strong isotope fractionation. I mean, for carbon or sulfur.
I'm not sure actually how they affect. If they are involved to that degree in this in the fractionations. Maybe. I haven't heard of it. But they exist there.
Okay. I asked this just because of COVID. Realized that how
this study came out just this study of my colleagues. It came out just in the times of COVID when it was the most COVID, and everyone said, "Okay, did you just dig up viruses from the deep subsurface in these times?" Just as a joke, because this is the worst thing. Are you now finding new viruses that we didn't know? Who knows? I mean, a lot of these systems down there, it might have possibilities that we didn't expect also, such as fungi. They are degrading organic matter and they're producing biomass, such as a bioreactor, but maybe they are much more effective than any fungi that we know of, because we don't really know what are these fungi and how do they work together with the methanogens. So it's a lot of work to be done on cultivations, for instance, of these systems that I'm looking into at the moment.
Okay. Actually, another relevant following question is that for this deep life, actually, in one of the slides that you show that the carbon isotope fractionation has larger variation. Like I said, 200, 300 meters, that was probably the largest. And then it narrowed down, and likely that indicate a decrease of microbe activity. So,
yes.
So I know that how does the energy system that supports? Because to me, that for someone that works on high pressure experiments, that pressure is not a problem for deep life. Sharman showed 20 years ago that E. coli could survive at 2 gigapascal condition. And to me, that temperature, okay, I buy it to 120 degrees C. That's what currently people see, but and the high pressure condition that might be further extended a little bit. So other than that, my curiosity is that, okay, how does energy system that really sustain it? Because even with the appropriate temperature or pressure, as well as water that is available, but we still need this energy system that to support it. So may I know that, I say, from your perspective, how does this works out?
Yeah, I mean, what you find in this plot that we go down to 800 or 900 meters, this is a fossil system. This is something that occurred maybe 400 million years ago, because we had then had 3 or 4 kilometers of sedimentary rock also on top. And we look at the fractures. But when it comes to high pressure systems, I agree with you that it probably could be even higher temperatures than we know of, but there are a lot of systems to still study. But this is the highest that have been found at the moment. But they are also extremely slow in these systems, so it could be maybe that they can persist even better when they are these extremophiles than from other systems that have more energy, because they are used to work with so little that when they get something they can turn on. It's like almost like a TV. You have it in standby and then someone comes and turns it on. It's almost the same with these microbes, that they are just sitting there almost dead, like zombies. Mm-hmm. And then a little bit piece of energy comes by, or the conditions are better, they switch on. Mm-hmm. So maybe this could also be done at even higher temperatures than we know.
Mm-hmm. So the energy provider molecules or compounds that do you suggest that they are the, let's say, hydrogen, methane are the major ones, or do you think that there are others like sources of this, let's say?
As we know, our organic carbon decrease very quickly in the upper part. Then it's left with what comes from radiolysis, for instance, hydrogen gas, can be methane. It can also be microbial metabolites themselves, dead microbes that are recycled in the system and becomes food for other microbes. So in some systems, the deep systems, most of the biomass or most of the organic carbon that floats around at 3 kilometers is actually microbes themselves, or dead microbe pieces of organic carbon that comes from some dead microbe, and not organic matter from the surface, because this is very fast becomes eaten by microbes in this near surface area. And then what is left is something that is very refractory and cannot be easily degraded. So someone eats the easy parts first, very quickly. Then the rest takes time.
Okay, that sounds cannibalism. But it's interesting because in my previous conversation with Jurinsky at the Johns Hopkins University, because they are group focusing on the equation of water and the high temperature, high pressure conditions. And I forgot the details he was mentioning about, but he was talking about the limit of how this possible organic compounds that could be saturated or dissolved in the water under this high pressure, like relative high pressure, high temperature conditions. Although he was talking about some other subduction zone, etc. But I think that's also relevant to life. Okay, there is one question from a participant. Ben Ching from Zhejiang University is asking, he's curious about the extreme negative carbon isotopic composition produced by microbial. So whether it's reasonable since the plants can fractionate the carbon isotope because there's a relatively long time process such as a photosynthesis. So how does this work? Is it reasonable for this very extreme carbon isotope fractionation to occur? That is very. So how does the anaerobic process that could generate this very extreme carbon isotope results?
First of all, we don't know because we haven't seen it in the laboratory. No one has been able to form these isotope signatures in a carbonate in the laboratory. So we cannot be sure. But one thing that we think of is, of course, that it's methane, microbial methane, that this becomes very isotopically light. This we have done in the laboratory, and this we know. This is the carbonate reduction pathway that creates this lowermost delta carbon 13 values. And if you have only methane in the system, and then you anaerobically oxidize it, then there is also fractionation in this step. So if you have a methane that is at a -80 or -100, and you fractionate it, or you use it, oxidize it, then the bicarbonate could be even 30 per mil lighter than the methane. And if you only have methane in the system, you can end up with a calcite with this composition. In marine sediments, the usual case is that the carbonates are heavier than the methane. And this is basically one idea, is that we have much more organic carbon in the system that dilutes the signature that comes into the calcite. But it seems in this case, there is only one process, the process of microbial anaerobic oxidation of methane that is in itself microbial. This is the most straightforward explanation to get these extremely light values. And when it comes to the extremely heavy values in the calcites, plus 37, it can also be the same, that we have a huge fractionation when we form the microbial methane in the first place. And we end up with a CO2 that is very heavy. And then this becomes calcite. I don't know if that answers the question, but this is how I interpret it.
Okay, I think that's so much about it. Thank you, Henrik. I'll keep you for an hour. And thanks everyone for your participation. And thanks, Henrik, again, for giving us such a great talk. And our next speaker will be Oliver Strauner. Actually, we are talking something from really deep, including the deepest flowing water people probably have known so far in Earth, all the way to 600 kilometers revealed from those deep diamond inclusions. Because they find some ice crystals from those deep diamonds that people calculate the temperature pressure and find that, okay, those ice should be flowing water in Earth transition. So next talk will be Oliver talking about that. And thanks everyone for your participation, and thanks our speaker again. So, okay, Henrik, bye-bye.